So far, we have been looking at the structure and functions of some biomolecules, such as carbohydrates, proteins, and lipids. Today, we are continuing with the structure and functions of nucleic acids, which are repositories or storage and transmitters of genetic information. So, in other words, nucleic acids store and transmit genetic information from parents to offsprings. These nucleic acids are divided into two types, being the ribonucleic acid and the deoxyribonucleic acid. As we continue, we will see the differences between these two types of nucleic acids. Then we'll also see the structure of nucleic acids being polymers of nucleotides. So we'll see the structure of nucleotides, the basic building blocks or the monomers of nucleic acids. Now, nucleotides are made up of three major components. That is the pentose sugar or the 5-carbon sugar, on which on carbon number 1 you have the nitrogenous base, and on carbon number 5 you have the phosphate group. Now we'll continue to look at the structure of each of these components. Of course, we had looked at the structure of the pentose sugar when we were studying the structure of carbohydrates. And of course, the phosphate group is an inorganic group, which is simple, and thus we will not be looking much into that. But let's go ahead and look at the structure of the nitrogenous base. So the nitrogenous bases are divided into two groups, which are the purines and the pyrimidines. And of course, we find the purines in both RNA and DNA, whilst for pyrimidines, you find thymine only in DNA, whilst you find uracil only in RNA. Looking at the structures of purines and pyrimidines, you see that the structure of purines is made up of 6 rings, which is the 6-sided ring fused with a 5-sided ring. But for pyrimidines, you find that the structure is made up of only a single 6-sided ring. Now, the differences you notice between adenine and guanine is that on carbon number 2, you find the amine group for guanine, whilst on carbon number 6, you find the amine group for adenine and the double bond O for guanine. When you come to the pyrimidines, you find that the differences between these two is that for cytosine, you find on carbon number 4 an amine group that you don't see in any of the other pyrimidines. Then you see that for thymine, there is a methyl group on carbon number 5, which also you do not find in any of the other pyrimidines. Now, when you come to uracil, you find that though you have the double bond O on carbon number 2, which is in all of them, and the double bond O on carbon number 4, which you find also in thymine, there is no methyl group on carbon number 5 for uracil. So you can notice these differences between the three pyrimidines, and you can notice the differences between the purines and the pyrimidines. Now, so what it means is that in an examination, when you see a 6-sided ring fused with a 5-sided ring, if the question is about purines, pyrimidines, quickly you know that purines will have 2 rings and pyrimidines will have a single ring. And you can pay attention to the differences in the structures of the various pyrimidines and the 2 purines to distinguish between these purines and pyrimidines. Now, we continue to look at the pentose sugar, which we had seen earlier when we were studying carbohydrates. And you see the difference between the sugar that you find in RNA and DNA. Now you see that for ribose, the ribose which you find in ribonucleic acid, there is a hydroxyl group on carbon number 2. But for DNA, you find that on carbon number 2, in place of this hydroxyl group, you have hydrogen. And so you see the name deoxy. Think of it as being that the oxygen on carbon number 2 is deleted. And so it's giving the name deoxy, without oxygen. So that is where the name comes from. Deoxyribonucleic acid, DNA, whilst RNA is what? Ribonucleic acid. So you notice these two, these differences, this difference between the sugars, the pentose sugar, which you find in RNA and DNA. So far you can see the differences which are between these DNA and RNA. When we're talking about the nitrogenous bases, we've talked about the fact that in DNA you find only thymine. In RNA you find uracil, which means in DNA, in place of uracil, you get thymine in DNA, and in place for thymine in RNA you find uracil. That is a difference that you find between RNA and DNA concerning nitrogenous bases. Now when you come to the sugar, the pentose sugar, you see the difference between the structure of the ribose and deoxyribose being on carbon number 2 of the sugar, where in RNA you find a hydroxyl group on carbon number 2, which is absent in DNA, in the deoxyribose. Now, before we look at the complete structure of the nucleotide, you need to distinguish between the term nucleoside and nucleotide. Now, you see that the nitrogenous bases, all of them, they have some shaded hydrogen, which is the reactive hydrogen, on the N9 for adenine and N1 for the pyrimidines. So for purines, you see that the N at the 9th position is having these shaded hydrogen, the reactive hydrogen, and this is what reacts with the hydroxyl group on the carbon number 1 of the pentose sugar to form a glycosidic bond between the sugar and the nitrogenous base. So in purines, the N9 with the hydrogen on it, this hydrogen is what is reactive to react with the hydroxyl group, giving out water, and then a glycosidic bond is formed between. Now remember, we said a nucleotide is made up of a pentose sugar, a nitrogenous base, and a phosphate group. Now, if there is no phosphate group, without the phosphate group on the fifth carbon, you have what is referred to as a nucleoside, not a nucleotide. A nucleotide will have a phosphate group, and so without a phosphate group, you have a nucleoside. Now we have just looked at the formation of the glycosidic bond between the nitrogenous base, which is a purine, and the pentose sugar. So let's go ahead and look at the formation of this glycosidic bond with the pyrimidine. So we see this is a pyrimidine, this is thymine, or this is uracil. Sorry, this is uracil, and uracil is bound. So this is on N1. Remember, the numbering for the pyrimidines starts from the N1 and it goes clockwise. So this is N1, on which you had the reactive hydrogen, which reacted with the hydroxyl group on carbon number 1, forming the beta N glycosidic bond between the nitrogenous base, the pyrimidine, and the pentose sugar. That is a nucleoside called uridine. Now, the phosphate group, which is attached on carbon number 5, if it is only a single phosphate group, then you have a monophosphate. So a nucleotide with only one phosphate group. So for instance, this is adenine. This base is adenine. And so if you have just one phosphate group, you have adenosine monophosphate. If you have two phosphate group, you have adenosine diphosphate. If it is three, then you have what? Adenosine triphosphate. Now, take note that the nucleotides that you find in nucleic acids are all monophosphates. They have only one phosphate group. Now, the diphosphates and the triphosphates are some form of energy, or some act as some secondary messengers, activators of enzymes. But in nucleic acids, the nucleotides are monophosphates. They have only one phosphate group on them. And of course, this phosphate group is coming from phosphoric acid. So that tells you that this phosphate group is what makes this molecule acidic. So as you will soon see, this phosphate group can donate protons or hydrogen ions in solution, making them an acid. Now, with that said, it means that if we take the bases or the bases which are present in DNA, so we are looking at the top here as all the nucleotides which can be present in DNA, and then below we want to look at the nucleotides which can be present in RNA. Now, at the top you find, yes, the bases that are present in DNA are adenine, guanine. These two are the purines that are present in DNA. And the pyrimidines that are present in DNA are thymine and cytosine. Now, without the phosphate group, you have nucleoside. And the nucleoside that is formed with the base adenine is deoxyadenosine. Remember, the sugar which is present in DNA is deoxyribose. And thus, you find that the nucleoside is what? A deoxyadenosine. For guanine, it is deoxyguanosine. For thymine, deoxythymidine. Whilst for cytosine, you have the deoxycitidine. Now, when you have the phosphate group attached in DNA, like I said, all the nucleotides in nucleic acids are monophosphates. And so then you will have a deoxyadenosine monophosphate. Now, the 5' that you see here is only indicating that the phosphate group is on the 5th carbon, as we saw in the structure earlier. And you see that deoxy is being represented with a small d in front when you look at the abbreviation of the name. So we have a deoxyadenosine monophosphate, and we have a deoxyguanosine monophosphate. All of them, you have the phosphate group on the 5th carbon, and thus you see the 5' being written. And of course, the D, you see the D which is representing deoxy in front of all these abbreviations. Now, when you come to RNA, of course, in place of thymine, you have uracil. That's the only difference. And then you see that the ribose sugar, because of the ribose sugar, you do not have the suffix deoxy in the name. You have adenosine, guanosine, uridine, cytidine. Then the nucleotides out of these, when the phosphate group is attached, then you have an adenosine monophosphate, guanosine monophosphate, uridine monophosphate, and cytidine monophosphate. Again, the phosphate groups are on carbon number 5, and thus you see 5' in the name. And you notice that in these abbreviations, you do not see the small d, which represents deoxy. And so when you see this AMP, adenosine monophosphate, clearly it tells you this is present in RNA. So you see all of these differences between the DNA and RNA, and even between the nucleosides and the nucleotides, all summarized in this table. So you can clearly see these differences even with the bases, the sugars, and the and you see that the differences between the bases, and then you see the differences between the sugars when you see deoxy for DNA, and then you do not have the deoxy, but you have ribose in RNA. You see all of these differences, and you see that even in the abbreviations. All right. Now, apart from this, we also have cyclic nucleotides, and these, you see the cyclic structure being formed intramolecular. So you find that for the phosphate group, which is on carbon number 5, this phosphate group can form an intramolecular cyclic group. So you see that this is carbon number 5, this is carbon number 3, and because of that, you have what is referred to as the 3' 5' cyclic nucleotide, because you have the cyclic structure forming between carbon number 3 and carbon number 5, on which you have the phosphate group. Now, these cyclic nucleotides are not found in DNA or RNA. You do not find these in nucleic acids, but they act as some second messengers in the activation of enzymes, in enzymes that are responsible for the transfer of phosphate groups from one molecule to the other. These cyclic nucleotides act as second messengers, the transferers of these phosphate groups. So you find the cyclic adenosine monophosphate, the cyclic guanosine monophosphate. The only difference in these two is the nitrogenous base. The cyclic adenosine monophosphate here is this nitrogenous base is adenine, whilst the nitrogenous base that you find in cyclic guanosine monophosphate is guanine. So you find these two as being activators of protein kinases. These are enzymes that transfer phosphate group from one molecule to the other, and these reactions are common in many metabolic pathways. Now you also find the 2' and 3' cyclic nucleotides. 2', 3' because you see that this is carbon number 2 and carbon number 3, between which you find the cyclic structure being forming. These are just intermediates in the metabolism of nucleotides. When nucleotides are being broken down along the line, you find that these intermediate structures are formed. So in reality, you do not have these cyclic nucleotides existing, but it is in the process of metabolism. You find them as intermediates in a metabolic process. Now, so if you find something like 2, 3 cyclic nucleotide, obviously these are not present in nucleic acids. So just as we saw with the 3', 5' nucleotides, we said these are not present in nucleic acids, but they act as some second messengers for the protein kinases, enzymes that are responsible for the transfer of phosphate groups from one molecule to the other. Similarly, you find the 2, 3 cyclic nucleotides also not being present in nucleic acids, but they are intermediates in the process of metabolism of these nucleotides. Then we also have the cyclic dinucleotides. So here is a dinucleotide because, as you can see clearly, this is coming from 2 separate nucleotides. So you see, this is a nitrogenous base on a pentose sugar with a phosphate group. So this whole structure here is one nucleotide, whilst you have another sugar, a pentose sugar with a nitrogenous base and then a phosphate group. This two is also a separate nucleotide on its own, and these two bond together to form a cyclic structure. As you see on carbon number 2, you find the phosphate group on this first nucleotide binding on carbon number 2, whilst the phosphate group on this second nucleotide is binding on the carbon number 3 of this first nucleotide, forming a dinucleotide. Now, these cyclic dinucleotides too, you do not find them in nucleic acids, but they act as some switches, some ribose switches of RNA of some bacteria. So they are responsible for various reactions, various biochemical reactions, various features that you find in some specific bacteria, which is responsible for their properties of virulence and even the formation of biofilms and even their movement. So you find these cyclic dinucleotides also acting as the second messengers in the reactions, in the biochemical reactions of these bacteria, but you do not find these in nucleic acid. So if you are reading biochemistry books and you find some of these cyclic nucleotides, the names, the structures, bear in mind that these nucleotides are not present in nucleic acids. As we saw earlier, the nucleotides which are present in nucleic acids, we discussed them. But if you see these cyclic nucleotides, they are not present in nucleic acids, but they have different functions. Now, we have talked about the nucleotides as the basic units or the monomers of nucleic acids. And we also said from the beginning that nucleic acids are polymers of these nucleotides. Now, remember when we were doing proteins, we looked at the primary structure of proteins being the sequence of amino acids in a polypeptide chain. Now, similarly, the primary structure of nucleic acids is the sequence of nucleotides that are in a nucleic acid chain. So what I mean is, if you take the structure of any nucleic acid, we said it is going to contain nucleotides as the monomers. But you see, all these nucleotides have the sugar, the pentose sugar, the phosphate group as a constant component between them. The only difference that you find in these nucleotides is with the nitrogenous bases that are on carbon number 1, if it is in RNA or if it is in DNA. And so these nucleotides are conveniently being represented by the first letter of the nitrogenous bases. So for instance, guanine is represented as G. So when you see G, really what it means is that is guanosine. When you see T, that is thymidine. When you see A, that is adenosine. Now, what it means is that you can state the sequence of the nucleotides in a nucleic acid chain by just using the letters. So if you see G, 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 it means guanosine, guanosine, guanosine being bonded. One guanosine bonded to the other, which is also bonded to another guanosine. Then it's bonded to thymine, it's bonded to guanine, it's bonded to adenosine, cytidine, thymidine, another cytidine. Now, when you are talking about the structure of nucleic acid, such that what you are referring to is the sequence of the nucleotides that are bonded one after the other. What you are talking about is the primary structure of nucleic acids. So this structure can be seen more in detail, where you see, say, this is adenosine, which you see the nitrogenous base adenine on the pentose sugar, and then there will be a phosphate group on carbon number 5, which is now bonded to another nucleotide. This is thymidine. The nucleotide is thymidine, so because you find the nitrogenous base thymine bonded on carbon 1 of the sugar, and the phosphate group on carbon number 5 is bonded to carbon number 3 of the other nucleotide. And you find this repeating. You find this repeating when you come to guanosine. The same thing you are seeing over here, that the phosphate group on carbon number 5 of one nucleotide is bonding on carbon number 3 of the other. Now, these bonds that you find between the nucleotides that are bounded together in a nucleic acid chain is referred to as a phosphodiester bond. Now, phosphodiester bond, because you see the ester bond when you were doing the organic chemistry, you see that you have COC, carbon oxygen carbon bond, being referred to as an ester bond. Here you see it is phosphate oxygen carbon, and you see it repeating also vertically, where you see carbon oxygen phosphate. So if this is one ester bond and this is a second ester bond, then that is a diester, and this diester is being formed with phosphate. That is why it is being referred to as phosphodiester bond. So remember, the bond that exists when we were doing carbohydrates between monosaccharides, we said the bond that exists was glycosidic bond. When we came to proteins, we said the bond that exists there is a peptide bond. Here you see the bond that is existing between the nucleotides in a sequence is a phosphodiester bond. Okay, now, so again, we mentioned earlier that the phosphate group is responsible for the acidity of the nucleic acid, because it is able to donate a hydrogen ion. So you see these hydrogens on the phosphate group shaded. It is these hydrogens that are donated as protons or hydrogen ions in solution, making this phosphate group then to carry negative charges. And these negative charges help these nucleic acids to bond with metal ions such as sodium and potassium in solution, and so they can form the salts of these nucleic acids. Now, the secondary structure is what is referred to as the double helix. So you see, when we were looking at the chain, the long chain, the chain of a nucleic acid, we're talking about the phosphodiester bond between the nucleic acids. Now, when you come to the secondary structure, where you have two nucleic acid strands now bonding between the two of them, and this bond is existing between the nitrogenous bases that are in these nucleotides. The bond that exists between these two strands in DNA is the hydrogen bond. So you see that the hydrogen bond is what binds two strands together in DNA, but the phosphodiester bond is the bond that binds the nucleotides in sequence in a nucleic acid chain. Now, these nucleic acid chains that are bonded by the hydrogen bond between them, you will see that these run anti-parallel. We'll soon see what that means. So you see the primary structure being the sequence of nucleotides, and you see the secondary structure being what is referred to as a double helix. Think of it as a ladder which is twisted, or it's like a screw or two ribbons that are wound around each other, and it has these hydrogen bonds between the two strands. Now, the bases that are having these hydrogen bonds between them are referred to as complementary bases. Now, this is based on what is referred to as the Chagall's rule, which states that for each adenine in a chain, you have a thymine that is opposite to the thymine that is bound to it. So you have adenine, which is a purine, and to that adenine, the complementary base is thymine, which is a pyrimidine. Similarly, you have for guanine, which is a purine, its complementary base is cytosine, and vice versa. Now, the differences between the hydrogen bonds between these two complementary bases is that you have thymine and adenine having two hydrogen bonds between them. But for cytosine and guanine, you have three hydrogen bonds between them. So you find cytosine and guanine being complementary bases, such as thymine and adenine are complementary bases. In other words, the number of adenines that you have in a nucleic acid is equal to the number of thymine that you have. Similarly, the number of cytosine you have in a nucleic acid chain is equal to the number of guanine that you have in a DNA molecule, because these are complementary bases. They bond to each other. Now, since we talked about the fact that DNA and RNA, nucleic acids, are repositories or storage of genetic information, and they also transfer this genetic information from parents to offsprings. We'll briefly look at the overview of this process of transfer of genetic information, where you see DNA and RNA acting. Of course, we will not be going into detail of each of these processes. At our level, we are just looking at the overview of how this transfer is done. If you get interested and want to continue to do BTech, SLT, BTech, MLT, we go into detail into each of these processes. Now, what you see here in the transfer of genetic information is what is referred to as the central dogma of molecular biology. And this is about the fact that from DNA, RNA is produced, messenger RNA is produced, and from the messenger RNA, proteins are synthesized. Now, so if you have a DNA, which is a double strand, and this information which is stored in DNA needs to be copied first of all onto a messenger RNA. Before that, this DNA would have to split into two before it can be copied, and that process is what is referred to as replication. So the DNA is split into two strands, opening up for it to be copied. And the process of copying that genetic information from the strand is what is referred to as transcription. And you have the messenger RNA being copied from the DNA. So the information from the DNA is being carried on a messenger RNA. And this messenger RNA now binds with a ribosome. Then a transfer RNA, which is bound to some specific amino acids, now come onto this complex of messenger RNA and ribosome. And you see that a peptide chain is being synthesized from this complex, because each of these carrying an amino acid will be attaching one after the other to form a polypeptide chain. Then you have the protein produced from that DNA. In a sequence of these processes: replication, transcription, and translation. Now, it might be difficult for you to understand this process because, of course, you are looking at it just on a flat screen. I will try as much as possible to get you a video that you watch to understand these processes of transcription, translation, and even replication, so that you see this in a 3D. Of course, we are only looking at the overview of all these processes one after the other without looking much into detail of what happens, because like I said earlier, we'll look much into detail of each of these processes, not at the diploma level. When we are doing, say, when you enroll into BTech, SLT, or MLT, we will have that. All right. Now, so based on that process of transcription, translation, where you have the proteins being produced, you need to understand that each of these nucleotides in triplets correspond to a specific amino acid. You see, when we were doing proteins, we looked at 20 amino acids which are found in proteins. Now, each of these amino acids will correspond to triplets of nucleotides. So, for instance, the triplets uracil, 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 or uracil, uracil, cytosine will correspond to an amino acid phenylalanine. This other one will correspond to leucine, and so on. Proline, threonine, valine, alanine. This is asparagine. This is aspartic acid. This is glutamic acid. This is glutamine. This is histidine. This is tyrosine. So you see serine, arginine, all of these triplets are corresponding to an amino acid. You see this red one, which is AUG, meaning adenosine, uridine, guanosine. This sequence of 3 nucleotides together, this triplet, corresponds to the amino acid methionine, and it is always the first to start the building of, or the synthesis of, a polypeptide chain, and this is referred to as the start codon. So at least you don't have to keep all of these in mind, but remember that AUG is a start codon which corresponds to the amino acid methionine. In other words, the synthesis of proteins will be starting always with the amino acid methionine. Whilst stop codons, so the stop codon, so what it means is stop codon does not correspond to any amino acid. It signals the end of the synthesis of a polypeptide chain. So uracil, this is a uridine, adenosine, adenosine, stop. Uridine, adenosine, guanosine, it's also a stop codon. And then you have UGA, also a stop codon. You have 3 stop codons, and you have only 1 start codon. These you can keep in mind, because a lot of times you have biochemistry questions on start codons and stop codons. There is only 1 start codon, and there are 3 stop codons. You can keep that in mind, but not all these others. So you see you have 61 different codons for amino acids in these, what is referred to as the genetic code. So this is how information is stored, this genetic information. Remember, these proteins which are synthesized, when we saw we studied the structure and functions of proteins, we talked about the fact that the proteins can become enzymes, they can become hormones, they can become neurotransmitters. So depending on the protein which is synthesized, these genetic codes are responsible for the various sequences of the of the various amino acids, the sequences of the various amino acids which will come together to form a specific protein, which can become any of those biomolecules, either an enzyme, a neurotransmitter, hormone, etc., taking part in some specific biochemical reactions. So on this note, we end with nucleic acids. Of course, when we meet in class, we are going to look again at nucleic acids, the structure, the functions, face to face. If there are any questions, I will answer them. If there are any contributions, anything that you noticed that you didn't understand well and you want some more clarification, we'll look at all of those. So all the best. We have come to the end of this lecture till we meet face to face the next time.